Thank you, Banked, and thank you for the invitation to be here. I'm going to talk about a subject that's a little bit different than I usually talk about, but the techniques are the same as that I usually talk about. And I'm not an expert in learning and memory, but I've never been afraid to ask questions in areas that I'm not an expert and then to learn, okay? And I think that's a very good thing to state up front, both that I'm not an expert and it's always good to look at things that you're not an expert in and then learn. I'm going to talk a lot about dopamine, which is, I hope, appropriate following the great father of dopamine, Arvid Carlsson. I'm going to talk about it more as a tool. I mean, it's a wonderful molecule because, as Professor Carlsson said, it's used for, it's very easy because you can identify its effects, but it's also wonderful because it's easy to detect, and so analytical chemists have loved it for a long time and have helped in that citation index, I'm sure. I'm going to give a simple hypothesis in learning and memory, and I'm going to talk about lipids. Arvid talked a lot about receptors and so forth, but lipids I call the forgotten molecules. And very much like what you said in the beginning of your story, for the last 15 years, people have sort of told me that I'm crazy. In the beginning, people would say, why are you looking at lipids? It's all proteins. And proteins are very important, okay, no question. But lipids are important too. And when you see microstructural changes in the brain, we've imaged some of these with drugs. And I won't show this, but the fact is that the lipid composition and the distribution changes when you give drugs. And this is really going to be important too. And I'll come to this later on. I'm going to talk about two major methods from my laboratory, electrochemistry and mass spectrometry imaging at the end. And so I'm going to teach you what these are for all the students that are here, very briefly, but in very simple terms. And I'm going to try to keep the whole thing as simple as I can. If I start speaking too fast, because many of you English is your second language, raise your hand and say, hey, you're going too fast. Okay, that's okay, all right? Maybe I won't finish, but that's okay. Please do that. So the lessons that I'm going to show you in the talk today are, is this right? Are one, that open and closed exocytosis is normal exocytosis. For the neuroscientists, they know what I'm talking about here, but everyone else probably doesn't, and I'm going to show you what I mean by that. And that exocytosis doesn't happen the way that we think it does. Okay, at least in the textbooks. They're being revised now. Kandel's most recent book is revised at some. And I'm going to show you that small lipid changes in the membrane can make big differences and might be part of the initial memory formation, what I call short-term memory. And it fits with what people like Kandel have been saying about memory for a long time, okay? And I'll show you that as well. So let's start with the neuroscience. And this is an old Scientific American article, Neuron. Doesn't look the same on this screen. But this is, the nerve cell has a cell body with an axon, or axons that come down, usually one that comes down to a lot of terminal fibers. There can be thousands of these connections to the next neuron. And at these connections where the terminal fibers come down, they form synaptic boutons that in cartoons look like this, okay? In reality, they're a bit different, but it's good to understand. And most of the pictures you'll ever find in the literature and certainly in textbooks have these vesicles inside of these synaptic boutons, these ends of the neurons, filled with neurotransmitter molecules. These are these little balls in here, okay? They're supposed to be molecules, and I'll show them in a minute. And when the nerve cell gets an action potential or a signal from this part down to this part, it wants to signal or communicate with this part, and it releases these neurotransmitter molecules by causing these vesicles to come down, dock with and then fuse to this membrane of the end of the cell and dump out their contents, which go across and interact with these receptors that Professor Carlsson was talking quite a bit about. This is very important to understand this. And for decades, neuroscientists have basically thought that this was an all or none process. That the real process was on how many of these happened, not what happened with these individually. And if you think about it carefully, you realize that that's probably not logical, but we'll go into that maybe in a little bit. And if anyone wants to ask me, I can talk about this more later. But we've been studying this process of exocytosis in quite a bit of detail, and in our systems, for the most part, these little molecules are dopamine. Okay, so there's some consistency with what we're talking about. So when you think about exocytosis then, the vesicles are made of a lipid bilayer, it's shown here, and these are molecules with long alkyl side chains, okay, or tail groups. These are carbon and hydrogen chains that have head groups that are more polar. So they like water, and this doesn't like water in here, and this likes water because you have head groups on the inside and on the outside, and then you can store molecules like the neurotransmitters inside of these vesicles where there's also some water. Not a lot, but some. Then there's also proteins in here, and I just make them a big black ball. Okay, people that know there's more detail. This is a dense core. We're looking at what we call dense core vesicles because there's a lot of protein in there. And there's a lot of interest in what happens here, but I won't talk about that today. The vesicles, when you want to communicate, when the one cell wants to communicate with another, they come up and they first form a pore like this, and then it's thought that this opens up, as I was saying. But instead, what we think happens is that they close again after some neurotransmitters come out. So what you can think of this is like this vesicle comes up to the surface, it's got liquid inside with the neurotransmitter molecule, and it's like a squirt gun, and it goes, squirts a little bit and closes again. And I would say our data, and I won't go through all of it, but I'll give you quite a bit of it, says that about 95 to 98% of the time, that's what happens. And maybe about 2 to 5% of the time it opens all the way. Maybe when a vesicle needs to recycle, it's lost some proteins, then somehow there's a mechanism in there that allows it to open all the way. It goes over to where the coated pit is, this thing that helps you recycle it, pulls it back in again, and you start over. Vesicles are enormously complex. There's hundreds, if not thousands, of proteins on these tiny little structures. And there's been some beautiful articles showing the pictures of these cartoons that make them look so complicated that you realize it would be silly for biology to waste them with one shot. All this work to make the vesicle, and then poof, it's gone. That would be silly, okay? And most of us realize that evolution is not silly. It's very well structured. And so this is what we think happens. If only a fraction comes out, you have to ask some questions, okay? This is important because it can be regulated. So now this becomes a pharmaceutical target, okay? So drug companies I've talked to are pretty interested in trying to understand this better. It's also regulated. In regulating it, it means that individual events could be involved in synaptic strength, as in short-term memory, okay? Changing this. What Professor Carlson talked about as plasticity, okay? Changing from with a stimulus, a change in the event. That's what could be happening here at the single vesicle level. Very important, actually. It also, on the side, could explain the differential release of peptides and catecholamines. And peptides co-mingling with catecholamines has been a big issue in Sweden for a long time with Hochfeld, et cetera. Okay. Quantifying release now. Let's go to the first method that I want to talk about. So I want to show you this exocytosis event. I want to show you how we can measure it. And so we take carbon fibers. Now, carbon fibers are used in everything from airplane wings to brakes to golf clubs, okay? They're used in all sorts of things because they're resilient. They're very small, and they use them in big bundles and these other things. We take 5 micrometer diameter carbon fibers, and we seal them in glass, polish down the edge, okay? It's a very microscopic thing, but it's not so hard. There are tools to do this. And then we can take this and under a microscope, lower it down onto a cell. Now, in the cells in culture we use are pheochromocytoma cells, or PC12 cells. They're derived actually from the adrenal gland of a rat, and it was published in 1976 for the first time by Green and Tischler. So they've been around for a long time. They're immortalized cells, so they're cancer cells, and so we can keep propagating them forward. And if you have the right cell line, which is a trick to this, you can actually get these cells when you stimulate them with something like potassium chloride. Potassium changes the potential field across the membrane. You stimulate them with potassium chloride, they'll undergo this exocytosis process on the basic undifferentiated or the round cell. Okay? And if we put the electrode right on top, just trapping a couple hundred nanometers of solution perhaps on top, you can see this event coming up one at a time. We stimulate the cell, you can see it because at the electrode we can oxidize dopamine to give us dopamine orthoquinone, and when we oxidize it, we take 2 electrons. That gives us a current, okay? And the number of electrons, we can measure that in terms of current, is proportional to the number of molecules. So when we do this experiment, we put the electrode here, we stimulate the cell, we stimulate at each of these arrows, and this is a 30-second bar, and this is 175 picoamperes, so there's a lot of experiments because they're very fast, as I'll show you in a minute. We stimulate, you see there's a bunch of current transients, or peaks, spikes, we sometimes call them. Then it slows down. Then we can do it again, and then it slows down. The cells don't like to keep releasing. They do it for a little bit, then they equilibrate. If we blow one of these up here, 10 milliseconds by 50 picoamperes, we can find that this is what it looks like. It goes up pretty fast, comes down a little slower. Okay, so we have this spike. This spike corresponds to one exocytosis event. We're measuring the molecules coming out by looking at the number of electrons. If you integrate the current, you get the charge. And the charge is directly related to the number of electrons, which is related to the number of molecules. Pretty simple, right? And so there's this wonderful equation called Faraday's equation. So you students, I mean, you often get really complicated equations. I hate them. I look at complicated equations and I go, I'm asleep, okay? I can't deal with them. But this one, I can deal with this one. Well, I even memorized this one, okay? All right, so it's N is the number of moles, okay, of dopamine in this case. Q is the charge, integrated current. N, little n, is the number of electrons, and F is this Faraday constant that relates the number of moles to the number of electrons. Very simple. So Q over NF gives us the number of moles that are coming out. And we were doing this experiment for a number of years. We started this in 1994, and Whiteman's group did it on adrenal cells a little before we did. And what happened here was people kept asking me, well, I would relate to what came out to what was in the vesicles. And people kept asking me, how do you know everything comes out? I said, oh, well, it's in the textbooks. Everybody's sure it all comes out, right? It's not true, okay? We went back and we started saying, let's develop techniques to prove that it all comes out. So this is a case where I wanted to prove something, and I disproved it, okay? So what we did is, after about 5 different failed attempts over many years, okay, and this is important, I should point this out. A lot of students started trying to do different techniques and gave up. And I have students now trying to replicate or invent new techniques that I'm not letting give up, okay? Some are in the audience. And so what we did is we came up with a technique where we take a capillary. Now this capillary has like about 15 micrometer inner diameter. It varies up and down from there. And it's a very small inner diameter. It actually goes way over here, all right? And we do an experiment called capillary electrophoresis, which is just separation in a potential field. It's nothing more than that. Things with different charges and different sizes will separate out. That's all you need to know there, because I'm not going to go into details. And when we do this experiment, another guy named Edgar Arriaga at Minnesota showed in about 1999, showed that if you put liposomes on these things at one end and apply this potential, they would come out the other end one at a time. Boop, boop, come out this way, right? Very nice. And so what we did was we thought, after these other failed attempts, we thought, well, why don't we try this method and see if we can do it with our vesicles? So we take the vesicles from inside of cells, these things that hold neurotransmitter, we put them on one end of the capillary, we run them down here, and they come out more or less one at a time. Then when they come out, we put the end of the capillary inside of a device we call a microfluidic device, where we have made of a polymer, and we put a buffer in here that causes the liposomes to start to destabilize. And when these liposomes destabilize, the buffer also focuses them onto a carbon fiber. Now, instead of a disc, it's a cylinder, so it's longer, 100 to a few hundred micrometers long. And these vesicles, when they hit the carbon fiber, they should lyse, and then we can do this electrochemistry experiment again to detect the dopamine, okay, if there's dopamine in them. And so here's the electrode. Here's where I'm talking about these vesicles with neurotransmitter come out. If they hit the electrode, they should lyse on the carbon, because these lipid structures don't like carbon, and we've got a sodium dodecyl sulfate buffer in here that also they don't like so much. And if they hit right at the time they lyse, we should detect what's in them with 100% accuracy, or nearly 100% accuracy. If we do this, what we find is we get a current transient that looks something like this. Okay, so here's time, here's current. It looks just like what I showed you for the release, except now we're looking at the entire contents of the vesicle. Okay, very important to realize that we're looking at that. Same equation. You integrate the current, you get the charge, and the number of moles equals the charge over the number of electrodes over the Faraday constant. We can tell how many molecules, but now instead of what's released, we're looking at what's totally in the vesicles. Okay, so we can compare. All right, so we did this. If we do the if a vesicle lyses out here, realize you probably have the question. If a lot of you like to ask questions. You say, what happens if it lyses out here? Well, if it lyses out here, diffusion's in 3 dimensions. Even if it's about 200, 300 micrometers away from the electrode, you see nothing. Okay, it's orders of magnitude lower in concentration because it's in 3 dimensions. Diffusion is never, hey, let's go this way. These molecules can't get together and say, hey, let's go over to that electrode. Okay, it's always random. Okay, random walk, Einstein, 1905. Right? So PC12 cells, these cells that I've been talking about, we had to isolate some vesicles, so we cultured them. We break them up, homogenize them. We do a hypoosmotic lyse. So you go to low ion concentration. That's supposed to keep the dopamine in the vesicles. If some of it leaks out, what I'm going to show you would be even more pronounced, not less. So we're pretty safe with this. When you do this, this is a pretty cool picture I took off the internet. So it's just shows you the lysis of these cells exploding. Okay, looks like something from astronomy. And then we do differential centrifugation. So we spin the tubes with these vesicles in them, and we find a layer that's enriched in what we call vesicles. And they call it synaptic vesicles in the prep that we had from Sigma Aldrich. It's a real pain in the neck. They stopped making this and selling this, and now we had to replicate it. It took a lot of work. Okay, so then I had 2 people, a postdoc and a student, work on this side by side with sister cultures. So they looked at them identically. Here's the electrochemistry looking at release. Here's what I call the electrochemical cytometry, measuring the total vesicle content. Jan and Donna did these experiments. And we get these kinds of data, these data. What we have are individual events from the release, individual vesicles being lysed and detected the contents. The reaction, again, is dopamine going to dopamine orthoquinone. The equation, again, is Faraday's law. So we can compare the two, okay? When we do this, we plot the data in what we call a cube root plot. And this is because vesicles have a Gaussian or near Gaussian distribution of radius. Radius cubed gives you volume. The amount is proportional to the volume. And so if you take the cubed root of the amount, it should give you a near Gaussian distribution. And we like Gaussians. They fit on top of each other better than skewed distributions. You can also do a log normal, which is even a little better. But this is the way we're doing it here. And so we have the number of events over the total number of events, and then done by times 100, so it's percent. Okay, so we're down pretty low here in numbers because there's lots of them. And we then take all the events that are at any given size and we bin them into small groups. So here, this is about 3 zeptomoles cube root. This is about 4, etc. And we go through here. The average actually in zeptomoles for the amount released is plus or minus. It's about 100 zeptomoles. Okay, so it's a small amount. But this is, again, cube root. And this red here shows the released. The black here shows the amount in the vesicles. Now, the 2 distributions look close, but they're not, because it's a cubed root axis. Okay, this compresses the axis. And so if you look up here, actually, if for 946 release events, we had an average amount that was 141 zeptomoles. Now, remember, we invented the concept of the mole, so we didn't have to talk about huge numbers. Now we're down to few numbers, and we talk about very small moles, which is really silly. So we talk about 10 to the minus 21 moles. That's a zeptomole. But really, that's 85,000 molecules. Here, for about almost 30,000 vesicles we measured with the cytometry, it's very high throughput. They go through really fast. 387 zeptomoles, or 220,000 molecules. So we're talking about thousands of molecules. Now, these are bigger vesicles because they're in a cell line that's derived from adrenal cells. But it's still very nice to compare this. If we look here now, comparing this number to this, the average PC12 cell vesicle releases about 40% of its total transmitter load during exocytosis. And like I said, some of it might be leaking out, so it might be even less than that. All right, so that was our first indication of this. There were actually several other things that we ignored because we didn't want to believe it, but this is our first real proof of this. And there's other people that have been working on this, especially Christian Amator in Paris, but he's using models to show that the release events I showed you before could also be explained this way. And the 2 of us have been promoting this, and now people are starting to listen. Well, if we look at the electrochemistry experiment where we put the electrode on the cell, and we're measuring single exocytosis events, Okay? Now, this is a really interesting thing, and I'm only going to show the one slide, but I think you'll find this cool if you follow me through the logic. So here's where we see one exocytosis event going up and down. And there's 3 different examples. And sometimes before the event, you see a little bit of a rise and then it stops. And this, for a long time, by another Nobel Prize winner named Erwin Nair, was termed the foot. So it's just before the foot. And Erwin Nair is a really famous physiologist. He invented patch clamp, okay, in the early 80s. Got the Nobel Prize around the same time that you did, but I think 2 years before or after or something. I don't remember, but it's somewhere in there. And I don't remember years so well. That's a good thing. And so we see this pre-foot, okay? And the foot is thought to be the vesicle opening a little bit and then getting stuck there. And it's because these proteins are involved in opening, these snare proteins they call them, and they're stuck there, and it gets stuck before it can open all the way, everyone thought. And that's what they proposed. Well, when we realized that the vesicles were opening and closing, my student Lisa Melander said, well, if they open and close, shouldn't there be post feet? Now, shouldn't there be a foot on the closing sometimes? And so she went looking for it. And sure enough, sometimes you see this kind of thing, sometimes you see both. It's pretty amazing. To see both is a rare event, okay, because they're not so common. And we can only pick out the feet where they're flat. On pre-feet, you can, because it goes up so fast, there's different types. On the post, we don't trust it unless they're flat. There's only a few % that do this, but they do it. And when you get them both, it's really amazing because every time the pre-foot to the post-foot, which is really relative to the concentration, goes down, okay? Now, I have a lot of other data that I couldn't figure out how to get off of here quickly, so I put a Swedish flag over it. We are in Sweden, right? But I'm going to ignore that. But if you compare a large number of these events where you have pre- and post-feet, you can see it drops down. It drops about 25%. It's right consistent with what we're saying with the cytometry. Really cool experiment. So that's electrochemistry consistent with the cytometry compared to electrochemistry. What about cytometry on a mammalian cell? You have to say those are PC12 cells. They're a mammalian, but they're a cancer cell line and everything else. A lot of neuroscientists don't trust them. So we took mice, and we did this with Ann Andrews' group, who's now at UCLA. We pooled tissue from 3 mice to get lots of vesicles. And Donna Omiatek again did this experiment. And we did cytometry. So we ran the vesicles through this thing, and we wanted to see what was in them. And when we ran the cytometry, these vesicles from the brains of mice, and from us probably too, have much less. A very challenging experiment. To measure a picoampere with this kind of time scale is really hard, okay? Right, guys? Okay, we can't do it now, but we could for a while, okay? Very hard. You have to do it in the middle of the night when no one's walking around to vibrate things. 0.5 picoamperes, here's one of the events. But when she did this, and she pooled a bunch of events, she looked, and for about 8,000 vesicles per nanoliter of injection volume, she found that the average amount in these vesicles was whopping 55 zeptomoles, or 33,000 dopamine molecules. That's 3 to 10 times what's thought to be released. Okay, so it's way more. It might be only 10% being released per time in mammalian cells. In fact, we've gone on and done pharmacology on this with the drugs we heard about before, reserpine and L-Dopa. We can make it go in half with reserpine, 5 times as much with L-Dopa. It all works beautifully. But really the point that I want to make here is that only a small part is released. This would be 0.8 molar for a 50 nanometer vesicle compared to 0.1 molar reported based on release experiments. So there's a lot of possibility for regulation of release in each vesicle. Is this related to memories? I don't know yet, but we're getting there, okay? So let's go back to this. Exocytotic release, open and closed, right? As only a fraction is released, then this is important because it can be regulated. It's a pharmacological target, and is it part of short-term memory? I still ask the question, but I'm going to show you some things, okay? So let's look at the general theory on short-term memory, and I just took this from a paper I summarized with a couple of bullets from a Candell paper in 2012. Synaptic plasticity, or changes with a stimulus or with a memory. What you're listening to me now, if you're not sleeping yet, okay, is synaptic. You have some kind of changes that are taking place. This is called plasticity. More or less transmitter released after an action is what Candell described it. Okay, and this, whoops. The stimulus, my voice, a pinprick in your finger, you know, a shock if you're doing an experiment. These are affecting intracellular messengers like cyclic AMP. That's what's proposed. Cyclic AMP affects kinase enzymes that are part of regulating release. Okay? And more or less transmitter release tends to lead to longer-term changes in cell structures and transcription. Okay? This is interesting because this is what most people sort of feel. I mean, this is one of the things people feel about short-term memory. What we know about short-term memory could fit into a very small vial. What we don't know about short-term memory could fill the world. Okay? There's a lot we have to learn. That's what my point is. But let me reiterate this idea that more or less transmitter released is where he starts to then come up with these things. And these things all happen, no question. But how much, is this all of the story, or are there other things that are involved? And how much from each? Let's see, okay? But first, how good is your short-term memory? That should be is. How good is your short-term memory? Okay? What methods have I used so far? Can you name one? Cytometry, electrochemical cytometry. A little different, but it's good. Another one? Oh, come on. Short-term memory. Amperometry, right? Or electrochemistry on cells. Nobody's afraid. Everyone's afraid. Is exocytosis all or none or open and closed? That's an easy one. Open and closed. Thank you. Okay. You know, how many people know what the rough attention span is for most humans? Very close. 10 to 12. Okay? 10 to 12, okay, is what they say. So in a talk, if I go for 40 minutes straight, you're going to just fall asleep. So every once in a while I have to do something silly. But this is actually, I think, important. Okay? All right. Lipid changes and exocytosis. A memory. Is it a memory? Is this part of memory? Let's think about vesicles. Here's the vesicle in this diagram I keep showing you. And in these junctions, we've been studying in other methods that I'll mention later, the lipids, if they're different shapes, some of them have conical shapes and they fit real nice into this junction. Some of them are more cylindrical. Most of the lipids actually are more cylindrical and they don't fit very well. You can see that. And so we've been studying that. But vesicles actually have a lot more of these conical lipids and lysolipids with one tail than the lipid out here on the outside of the membrane. It's about 5 to 1, actually. And people have shown with other experiments that when this event happens, and they originally thought it was opening and dumping it out, but actually it's probably a diffusion through the membrane during this short few milliseconds. When this event happens, because these are so much higher in this junction to make it happen, a lot of these move up into here. So what happens, and we know this, so what you could say is this is changing the structure of the membrane because you had a release event. That's plasticity, right? That's simple. If you have 20 release events, you've got 20 times the change. 200, you have 200 times the change. Very nice thought, okay, that this could be part of this plasticity. Well, does it make a difference that these are out here in terms of exocytosis? Okay? Is it possible that endogenous membrane lipids and lipid domains are important in regulating the rate and fraction of exocytosis release? And can cells control release by changing lipid composition? Well, we don't know the last question, but I do know the other one, because we've done this with amperometry. And what we've done with amperometry is we've taken an electrode and placed it on a cell, and then incubated the cell with lipids. And so if we incubate with lipids that are more cylindrical, like phosphatidylcholine or sphingomyelin, see the cylindrical structure, or more conical, like phosphatidylethanolamine, there's differences before, which is shown with the red, and after the incubation, which is shown with the black. You see what happens? It gets longer, it gets slower in terms of what comes out. And in fact, the amount that comes out changes with PC. It goes down by about 25%. With sphingomyelin, it gets longer here, and it actually seems to increase the amount. PE, in this case here, doesn't change the amount, but it does change the amount, but only by a small amount. We're not sure how much yet, but it makes it considerably faster, the event. So these lipids are having a large effect on the exocytosis process, both in time and the amount. Okay? Very important to realize that. And so we asked the question then, okay, if this is, and this is about the level of the information people got with cyclic AMP when they first proposed it for memory, okay? It's interesting. How much does the incubation change the composition of the lipid in the membrane? Because we incubate with 100 micromolar lipid. Well, you know, lipids are hard to get in, so we thought this was reasonable. People asked, I asked, how much does this change? If it's a huge change, then it means nothing in terms of the real brain, because the real brain's not going to change it. Well, maybe with domains, but probably not. A small change would be consistent with what a cell might do in starting memories, okay, short-term memories. So what we did is we took this technique called time-of-flight secondary ion mass spectrometry, okay, and we use it to, it's a big machine, okay, and we take our cells and we put it in this machine and we can use this to look at the membrane composition on the cell surface, okay, to see how we change it. And so what we did was this technique causes an ion beam, see if it makes it work, to shoot out of a gun, and then our sample is down here on this surface. And when the ion beam hits the sample, see the different color coming out, it knocks ions out here, and these ions come up here, and based on their size and their charge, they have different times to get from here to here, okay, coming this way. And so we can tell what their size is. That also the number of molecules getting here tells us how many are there. So when we do this, there's a pretty light slide in here, but this is the intensity, so the number of molecules we're seeing. This is the mass to charge ratio, so the size over the charge, 100, 200, and right here at 184, very small peak, but it's separate from the others, is the head group for phosphatidylcholine, okay? This is one of the main lipids in the membrane that I've been talking about, and this head group, we can detect it using this technique right here. And when we do this, if we do this at one spot and then move it to another spot and then to another spot, etc., we can raster back and forth and we can come up with an image where this is the intensity scale. So this is how much is there. Red is high, blue is low. Background is water, so we don't see the 184. We're just looking at 184, this peak, and here's 3 PC12 cells, about 20 micrometers in diameter, and with about a half a micrometer or 500 nanometer resolution, we can detect changes in these lipids across the cell. Now, for the experiment I'm going to show you now, we're just looking at whole cells, but we wanted to see how much did the lipid change in these cell membranes with this incubation. Well, it's really hard to look at it directly because there's lots of the lipid there. And if it's a very small change, you will never see that. So what we did is we incubated with lipids that were deuterated. So we all know what deuterium is. And you can take deuterium and replace the hydrogens, 75 in the case of the PC and 62 of the hydrogens in the case of the PE. And you get a slightly different molecule. Different mass, okay, it gives us different fragment masses. And so what we were able to do, here's the mass spectra, okay, here, you can't see very much. This is really light in here on this projector, but this is for PC, the phosphatidylcholine, this is for phosphatidylethanolamin. The conical lipid, I can't see the peaks at all in here actually. The conical lipid for the PE and the cylindrical lipid PC, again here I can almost not see the peaks, but here there are peaks here for the deuterated PC and the deuterated PE. Okay, so you get the idea we can do the experiment, and sort of, this here. But right here, these are the mass to charges, nominal masses, so we took away the detail. All right, and right here, there's a mass that's common to both of these when it's deuterated, but not in the normal form. It's mass to charge 50, okay, and really 50.1, and we looked at this mass. Now what we did is we took cells then, and we incubated them with the lipid, either the PC deuterated or the deuterated PE, and then we took them and we put them in a special device where we could fracture them open and look at their membrane surface, and we detected how much of these lipids that are present for the deuterated lipids was there compared to how much of the normal PC. So we could see how much it changed. Started out it was 0, and then we could do it at different concentrations. And when we did this experiment, here's the deuterated PC. It's all these D's here instead of H's for the most part. Deuterated PE. Okay, here's an image of a PC12 cell that's been incubated. This is looking at the PC, And we looked at this here. We did what's called a region of interest. We take this region and compared it to this region to get our experimental difference. Here is PC by itself without all these lines, and here's the deuterated PC in this case. There's not a lot of it there. You can see that. It's only sprinkled in there, but it definitely is there, and you don't see it if you haven't incubated with a deuterated lipid. Okay, so this is the key slide, because we incubated with 3 concentrations of each lipid, 100 micromolar, 200 micromolar, and 300 micromolar. Same thing here. 100 micromolar is what we did the experiments with before with the amperometry, okay? And here's the relative amount for phosphatidylcholine and the relative amount for phosphatidylethanolamine. And you can see it goes up in both cases with the concentration. So that's rewarding. It tells us we got good experimental paradigm here. And if you look here, though, for phosphatidylcholine, it only goes up 0.5% at 100 micromolar incubation. It's a very small relative change. For phosphatidylethanolamin, it goes up a little more than 1%. Again, a small change. If you look here, PC is 39% of the total lipid in the membrane, so 0.5% is a 0.2% change in total lipid. 0.2% is all we need to get that large change in exocytosis that I showed you. PE is compared to PC, so 1.5%, and PE is about 0.6% change in total lipid. Again, a fairly small number. And so the conclusion we came to is that only a small phospholipid change is needed for a large change in exocytosis rate. So endogenous lipids could be part of short-term changes in release. Okay, that I add in. That's my hypothesis. What do we know about related to lipids and short-term memory? Well, I just pulled up some random. There's a lot of papers. Arvid, I loved your comment about serotonin. It's a mess. It's all over. It's so complicated. It's the same for lipids. There's all sorts of studies, and there's lots of snake oil salesmen, okay? People who try to tell you one thing or another. I had them in my office talking to me about omega-3 and omega-6 fatty acids. I think they're really onto something. But the studies they quote are really out there, okay? And so yeah, we have to see something. There are some studies that are real scientific, though. Cholesterol, omega-3 fatty acids, and triglycerides have been postulated to play roles in affecting cognition and Alzheimer's disease. This is a huge area now with lipids and Alzheimer's. People are jumping all over this, okay? The elderly and obesity, okay? This is one paper, but there's a whole host of them. I have them in because we were writing a proposal related to this in our imaging. Cholesterol and some of the lipids, okay? Studies on the association between diet and cognitive function suggest a possible role of dietary fatty acids in cognitive decline. This is one study that I found was pretty scientific, pretty well scientific. Lipid lowering drugs seem to lower the chance of developing cognitive impairment, okay? Drugs. Lipids appear to be involved in the cognitive decline during AIDS. This has been a big topic in the last couple of years. And there are hints that ADHD patients have different fatty acid levels. It turns out there's a number of studies now that are pseudoscience that suggest that ADHD patients or hyperactive kids have high omega-6 versus omega-3 fatty acids. And I can tell you when you give methylphenidate to certain brain systems, I won't go into detail on that, it changes the lipid composition. Very interesting. So what do I have to say here to end here, more or less? Relative quantification of membrane lipids and learning and memory, I still have the question mark. It appears likely that endogenous membrane lipids and lipid domains are or could be important in regulating the rate and fraction of exocytosis release, okay? And that cells might control release by changing lipid composition. We haven't shown the endogenous so far, it's just exogenous. It seems, though, to me that this could be a good mechanism to add to those postulated for changing synaptic strength to form short-term memories. The initial, immediate part of it. I'm really excited about this. I think this could be a big thing that people have overlooked. I want to thank, this is the group, the overall bioanalytical imaging sensors and cells group. I showed people as I went along in terms of who did the work. These people are former stars, as I call them. These are people that are currently in the group. These are all agencies that support what we've been doing. And before I final end, I want to tell you about a cool book, okay? Because you guys might like this. If you're really a neuroscientist, this is too basic, maybe. Well, there's some things in there that I found pretty interesting. And I consider myself at least a fair neuroscientist. But this book by John Medina, and he's now got a second edition, called Brain Rules, 12 Principles for Surviving and Thriving at Work. It has all sorts of things. One of the chapters I find, I've been asked to lecture in groups that are non-scientific about exercise and the brain. We miss this all the time. I always like to talk to scientists and martial artists about sleep and the brain, because we all seem to forget about sleep, okay? Without it, if you sleep well, you memorize, you remember things better. Long-term memories are thought to be consolidated when you're sleeping, okay? Remember that. If you want to remember, you want to do well on your test the next day, you're better off sleeping than staying up all night to study. Memory, I separate out. Repeat to remember. If you want to change the CAMP, you want to change the lipids, you've got to keep doing it over and over, and then they'll change, okay? There's a lot of other cool things in here. He's written a whole book on children and the brain, brain rules for babies, which I found fascinating as well. But there's a lot of cool things here in this. And with that, thank you for your attention.